The good news is that you've gotten so much information about molecular biology that what I'm going to talk about is going to be very easy for everybody, so we'll fly right through it, I'm sure. So I'm going to talk about CHAMP1 and what we're doing at Mount Sinai, and the three folks in the back left should raise their hands because they've been seeing families all day today, and they'll be doing similar things tomorrow with CHOP. I'm going to get to biomarkers, but I'm going to put it in a larger context. One of the things, maybe this won't work. Say again? Oh, it did, yeah. So, I started out in genetics of autism, and one of the things that is kind of implicit in a lot of what we're hearing today is that as we look at these disorders, and this is autism, we find many, many genes that are mutated and cause that disorder, right? So you've heard about, this is a paper, so we run a very large international consortium with now 60,000 samples, 60,000 DNA samples. And you can see, for example, POGZ, I'll talk about ADMP. We're finding these genes that when mutated contribute to autism, intellectual disability, and developmental delay. This is, maybe I shouldn't point it on the screen, there we go. This is our most recent paper, and I can't make it out here, but ADMP, POGZ, many of the genes that I'll be talking about and that you've heard about are in these findings, right? And so we could take all of the genes we know about in autism and developmental delay syndromes and ask, if you go, for example, to kids and adults with autism, what proportion have a genetic mutation? And with current data, it's already 30%. We did this in an epidemiological framework, so we're very sure of that number. If you go to intellectual disability syndromes, it's even higher, 40 to 50%, and severe epilepsies is also very high. So we're now confronting these developmental disorders like autism and intellectual disability, language delay, motor delay, and realizing that they are many, many, many discrete genetic disorders. And that's a problem, right? Because it's hard to, you have to treat them, you may have to treat them individually. Okay, and so this is, Vanessa said earlier that you are not alone. This reality of many, many rare genetic disorders is clearly the case. Now, current estimates are there are 7 or probably 8,000 distinct genetic disorders. And how do we grapple with that? How do we develop treatments in the context of so many different genes? The other point that's been made earlier is that CHAMP1 is part of a larger complex, the HP1 complex. So this is, if you take this protein called HB1 and you ask who does it bind to, it binds to ADMP, CHAMP1, CDH4, CTCF, POGZ, and so on. These are, I picked those genes because all of those genes are involved in developmental delay syndromes. So it's quite likely that CHAMP1 and the CHAMP1 Foundation is not alone. That there is a family foundation for POGZ, there's a family foundation for ADMP, there's certainly a family for EHMT1. And so not only are there probably similar phenotypes, but it's also going to be the case, and it's been said several times before today, that it's very likely that pathways implicated in one of these disorders will also be implicated in the other, right? So, I got to point at the computer. Yeah, I have widescreen slides, so you're missing only about 20% on both sides. But if you look at our data, and this is the data that Kozo showed earlier from the Deciphering Developmental Disabilities study, many of the genes, and I can't make them out, but in this HP1 complex, POGZ, CTCF4, CHAMP1, they're all binding to the same partners. So they represent a biological pathway. And so we can learn about the pathway, and we can learn about, what you learn from POGZ will probably be related to CHAMP1 and vice versa. And in fact, there's talk about creating kind of a meta group of POGZ mutations, CHAMP1 and ADMP mutations, because there's probably going to be a lot of overlap in what you see, but also what might be effective in terms of treatments. Does that make sense so far? Feel free to interrupt. All right. So at the Seaver Center, we do a lot of gene discovery, and for a handful of genes, literally 5 of them at this point, we try and do 4 levels of analysis. And very briefly, we kind of study the gene target. We try and make human neurons with those mutations. We try and look at animal models with those mutations. And of course, we look at individuals with the mutations to characterize the phenotype. So you've heard a lot about animal models, you've heard something about iPSCs, you've heard about cellular work and so on and so forth. And the questions we can ask are, what does the gene do in cells, in Petri dishes? How do the mutations affect that role? Does it suggest something for treatments? How do neurons, what does the gene do in neurons? And what do the mutations do in that gene? So this is the stem cell work you've been hearing about. And then, can we take those stem cells, those human neurons, and can we use them to test a drug, right? And so you've heard about that too, right? If we had, with Searchlight and with Hakan putting cells in and making iPSCs and the work you heard about from California, if we can make neurons with phenotypes, you could put a drug on it and can you reverse the cellular phenotype? Genetically modified rodent models, you heard about a couple of mice already. Again, you can study the mice, understand the phenotype. What does CHAMP1 do in the mouse? And then, is there some treatment, as Hakan was pointing out and others, that can reverse that behavioral deficit? And ultimately then, what do you see in individuals with CHAMP1 syndrome as clinical endpoints, and then how do you actually address those clinically? So we take these 4 levels. And I'm going to talk a little bit about some of the cellular work, which is just ongoing. Matt Lally is a very gifted instructor at Mount Sinai, and we've gotten funding from the Brain Foundation. And the study we're doing here is we're doing large-scale knockdown of genes. So there's right now about 250 strong autism genes and about 500 strong intellectual disability genes that we know about. CHAMP1 is, of course, amongst them. It's going to be very hard to study one gene at a time, so we're trying to look at 50 to 100 genes at a time by using CRISPR to knock down all those genes. And I'm going to go back. Oh, no. Okay. Got to be here for the advancing. So this is a complicated slide, but basically these are the genes we knock down, and this is the measure of each of those genes. And what's on the diagonal in blue says, for each gene we knock down, sure enough, the expression of that gene went down, just confirming that we're making the gene go away. If we focus on, whoa, okay, I'm done, thank you, I'll sit down. Let's try, okay, we got to the very, very end, that's interesting. It's floating along by itself. Okay, so I'm not going to show you all 80 genes, but I'll show you genes in that complex. This is what we targeted. We targeted CHAMP1, and sure enough, when we measure CHAMP1 in neurons, it's down. We target ADMP, ADMP is down. We target CTCF, POGZ. So we're able to knock down specifically gene by gene and see what happens. We can look at in vitro different kinds of cells, precursors, neuronal progenitors, and glial progenitors. And the first thing we see is that CHAMP1 does not seem to change the makeup. So these cells now, these are nerve cells, and they start to differentiate into brain cells. And we can watch what happens. Does CHAMP1 knockdown affect how the cells grow into what they're supposed to be? And the answer is no. But we can also look at something called pseudotime. So these cells are developing in culture. They start out as primordial cells, and they make neurons with long extensions. And we're able to put a time point on the neurons. How far along have they been going? So this is pseudotime in this dimension here. So this is very early and later. And by the time it's in the yellow, you have really at the point of making neurons, nerve cells. And what you can see is that this is all those genes again, but look over here to the right, you can see that CHAMP1 in this assay, as well as POGZ, seem to push neuronal differentiation, so enhance the differentiation of primordial cells into brain cells faster than they should be going. You want everything to be properly timed. You don't want things to go too fast. So that is a cellular phenotype. That's telling you something about POGZ, and it may be something you can use to measure how Hakan's AAV vector works, somebody else's small molecule works, and so on and so forth. This is the same thing again, but basically showing that both POGZ and CHAMP1 seem to be pushing neurons to develop too rapidly. And there are gene expression modules, and here too we see that POGZ and CHAMP1 share modules. And, oops, I didn't realize this was going to be a, and of course we can look at differential gene expression, and we can tell you what's changing in these cells when we knock down CHAMP1. We can also see in the Venn diagram that there is some overlap between what changes with POGZ and what changes with CHAMP1. So that relationship is true on every level. What it does to neurons differentiating, what it does to gene expression, what it does to gene modules. We have a method called calling cards, which is a way of, well, complicated, but asking where CHAMP1 binds in the genome. You've already seen some work from Kozo about this. Basically, we see that CHAMP1 and ADMP seem to share binding sites in the genome, and we have a pretty good map of CHAMP1 binding sites overall in human neurons and culture. And interestingly, and this is a very important point about mechanisms, CHAMP1 appears to bind near its own promoter. And Kozo, do you see the same thing? Yes? No? Anyways. So that's important. Okay. So back to these four. So I've given you one level already, which is the cellular level, which is kind of molecular targets and human neurons. We aren't doing much right here with CHAMP1. But now I'll talk about what we do with families. And there was a white paper that just came out about gene-based therapeutics. And we use the word gene-based therapeutics as opposed to gene therapy, because gene therapy is one kind of way of attacking a gene or a gene product. You could attack the RNA, you could attack the protein, right? So a therapy that's based on what you know about the gene, the protein, the RNA. And there are specific challenges for gene-based therapies in the CNS. These are big ones, I won't lie. And then the ones we've already heard mentioning about timing. When does it have to happen for it to be maximally effective? And then, a really, really big one that pharma's thinking about all the time and won't even dip its finger in the water of a rare disorder is, how are you going to measure success, right? Nobody's going to develop a drug for X if X is not known, right? And so, how do you measure impact and success? Natural history, right? Well, see families, see them over time, see what's happening. Find endpoints and biomarkers. What is going to be the endpoint that you're going to use in a clinical trial? You say, if this changes, it's a success. Biomarkers is what I'll talk about a lot, which is, are there things that are easier to measure than complex behaviors that tell you you're going in a good direction? And then, of course, there are things like MRI. I mean, Håkan just mentioned that there are some MRI changes, structural MRI changes, I assume, in the mouse model for UC Davis. And then, of course, animal models. So these are things that, nobody can really say we're moving forward towards clinical trials if we don't have these measures. And so Tess Levy and team, Tess is the one on the left, on my left, on your right, we developed a new protocol. We had typically for rare disorders, the families come in and spend 2.5 days at the Seaver Center and go through a very, very detailed, painful, I'm sorry, it's burdensome, deep phenotyping, behavioral, neurological, and medical. COVID came along and we pivoted. A lot of people had talked about trying to do more of this work remotely because it's very expensive for a rare disorder for every family to come in, especially if there's only a couple of expert sites. It's also a question of finances and access. It's unfair to people who have less access. So, COVID gave us the opportunity to really push a remote protocol. And we decided, we were talking to Jeff, we decided that CHAMP1, we'd already done a lot of work in ADMP, we decided that CHAMP1 would be a good platform to test this. And so these are the tests that we did remotely. I won't go through them. These are some of the kids who came through and adults. Some of them are here, are here today. These are the mutations we looked at, and these are the other mutations in the literature. I won't go through that. But, we're beginning to map the syndrome, so we can talk about the Vineland, DP4, we could talk about the presence of autism, ADHD, we could talk about difficulties in diagnosing attentional deficits in the context of the specific disorder. We could talk about motor milestones, receptive language, and so on and so forth. So we're mapping the phenotype, but we're doing it remotely, which has some advantages and some disadvantages. And then we're able to now take all the data on CHAMP1 and begin to see what are common findings. Right? So we're beginning to develop kind of a prospective clinical picture of the phenotype. So what are Tess, Thariana, and Haley doing upstairs? They are doing the Vineland, some language measures, and the Sanford-Binet. I will say that there are different Vinelands. The Vineland we're doing is different than the one that you might be doing for Simon's or otherwise, and we think that this one's important, or important to do both. So the great news is that I think about 3/4 of the families we're seeing are repeat visits. So they were in the first study and now they're in the second study. Not a very deep phenotype, but we're getting measures over time, which is what longitudinal studies mean. So this really is the start of a longitudinal study. We'll be back next year, like that movie, I'll be back. So we can keep this going on. We'd like people to come to the center if possible. And then we're also collecting plasma in collaboration with CHOP. So this is some of the things that are going on upstairs from the original PV. This is language. And then I jumped onto, after we met with Håkan earlier last week, jumped on and we spoke to the Simons Foundation. We realized we didn't want to stick the kids 3 times to draw blood. Didn't seem like a good idea. We decided to work together. I went to Amazon and bought 2 different centrifuges. One of them is hand cranked. It's very exciting. It's upstairs now. And one of them, which is, we shipped them here. They arrived on Friday. And we're going to use them to do some of the separations we need to do for the biomarkers, which I'll get to in a second. So back to those back to the clinical side. But now to biomarkers. So biomarkers are, like I kind of intimated, they're ways of telling you something about a disorder that's not necessarily the final clinical endpoint. And there are many kinds. One of them is diagnostic. So a diagnostic biomarker for CHAMP1 syndrome is measuring genetics, right? So we already know about a genetic biomarker for CHAMP1. But there are biomarkers that are diagnostic, that they are prognostic, that can tell you about risk, prediction, risk, adverse events, and safety, and so on and so forth. And so a good biomarker for the kind of studies we're talking about, you already have the first and best one, which is the stratification biomarker. Somebody has CHAMP1 syndrome or does not. We're going to talk about treatments for CHAMP1 syndrome. You already know who has it and who doesn't have it. What you want now are, probably different kinds of things. Diagnostic, are there changes that tell you something about the syndrome? You just heard about mitochondrial potential. So if the blood changes that we just heard about implicate mitochondrial dysfunction, then you can think about mitochondria as a target, right? And predictive biomarkers are those that tell you, as this marker, this blood marker as an example, is getting better and better, it's going to end up suggesting strong improvement in the behaviors, right? And then if, for example, if there is some adverse event that happens, maybe a safety biomarker will say, this kid or this adult is, may respond badly to this treatment. Okay, so these are clinical biomarkers that we do a lot of work on, and we'd like to do also with CHAMP1, EEG, and this is a disorder called SHANK1 haploinsufficiency, and it is amazing. There's really no other example in neuroscience that I know of where you can see such a specific change in EEG, so traces on the brain, traces of neuronal activity, that is so consistent between individuals. So this is an amazing EEG biomarker for Phelan-McDermid syndrome. This is a social biomarker for Phelan-McDermid syndrome, and this is a sensory biomarker. So we like to do these kinds of clinical biomarkers as much as possible. This is that last one, the sensory assessment for neurodevelopmental disorders. Sensory issues are very common in these kinds of syndromes. If somebody's nonverbal, they're not telling you about it. We developed a way of observing individuals and determining their sensitivity to sensory, to visual, auditory, and tactile Symptoms is developed by Paige Cyper and Teresa Tabasoli. It's now being sold. And so, for example, back to ADMP, which is a syndrome that CHAMP1 and ADMP may have a strong relationship, and there are very clear sensory reactivity symptoms in ADMP syndrome. We actually think that could be a clinical target. I'll stop walking back and forth. And here is an efficacy trial, a safety trial, excuse me, of ketamine and ADMP syndrome, and some of the things we're doing upstairs were done there, but the sensory reactivity is one of the endpoints we used. And what you can see with a single dose of ketamine, looking at weeks, 1 week, 2 weeks, and 4 weeks later, let's see, sensory activity got better, kind of attentional deficits, hyperactivity got better, autism, social behaviors got better, and I can't see what I'm looking at, but other stuff got better too. So, you know, this is the first trial. It has limitations, but we can use some of those endpoints, clinical and otherwise, to kind of watch progress. So the other kind of biomarker is a molecular biomarker. And now that you know all about genes and DNA, you saw earlier DNA makes RNA, makes protein, and the proteins in your body make small molecules, metabolites. Citrate, what you heard about a moment ago, is a product in your blood that comes from many sources, including metabolism. So if we measure genes, we can find the CHAMP1 mutation. If we measure changes to genes, we'll get to that in a minute, measure RNA, we can see whether genes are expressed or not expressed. Protein, we can measure whether proteins are up and down, and we can measure the metabolic products of proteins. And we can do this all in the blood. Is it better to do in the brain? Yes, but the brain is not an accessible organ. Give you a couple of quick examples. This is a replicatable finding in SHANK3. We have a lot of individuals that we looked at. Everybody here is a unique person, and we can see, you know, many overlapping downregulation of multiple genes that are very consistent across individuals. We can look at gene expression changes, and those gene expression changes from the blood tell us that something was a little bit odd with the immune system. And so then we took blood cells and we actually confirmed that a certain subset of blood cells was changed in certain individuals with Phelan-McDermid syndrome. So we were able to find a new facet of the disorder by looking at gene expression in the blood. And this is these are the metabolites, the small molecules that we all have that are made by enzymes in our bodies. And again, there was a consistent, reproducible finding of changes in metabolites of Phelan-McDermid syndrome, which suggested that there was clearly reduced sphingolipid metabolism. We don't know quite what to do with that yet, but it's a biomarker. It might change as kids get better or worse, or adults get better or worse. It may also tell us something about targets. And this is another biomarker. This is epigenetics in ADMP syndrome, which is also called this very complicated name, Van der Haas syndrome, but it's ADMP syndrome. And we can actually look at different mutations, and they have a completely different, unique, unambiguous, completely replicatable epigenetic profile in the blood, right? Other groups have seen this too now. So you've heard about some of the discussion about, you know, what if all the mutations aren't acting the same? What if the terminal mutation at the very end is different from where they're missense? So here we see that any mutation in green in this gene looks completely different in terms of epigenetic profile in the blood. So it can also be a way of understanding subgroups of mutations. So that's all I'm going to say about blood-based biomarkers, except to say that we are collecting blood together with Håkan, and the idea will be to take that plasma and look at, I appreciate that enormously. Take that plasma and look at proteins, the metabolites, the proteome, the proteins, and at some future point, or ideally in collaboration with Håkan, look at RNA in the same exact blood samples, right? So that is our blood-based biomarkers. That is And the two examples I showed you with ADMP and SHANK3 have already shown highly reproducible and unique findings that we think are going to be important, we hope are going to be important to drive the field forward. And so we are hopeful, optimistic, that if we can do the same thing in CHAMP1, and we're not talking about thousands of samples, just like you heard with the citrate example, 20 or 30 could get you a long way down that road. So give blood. What do they say about voting in Philadelphia? Vote early and often. Is that about right? So give blood early and often. I'm going to stop there and thank you from the Seaver Center. Again, mostly I'm talking about what's happening now, what's going forward, but it's a pleasure. We like working with CHAMP1. We love working with the families. It's always great to meet everybody. So thank you. I'm going to stop there.
Yeah, super grateful for you being here and giving the presentation. It was awesome. I know I have a couple questions. We'll try to go as quickly as possible because we're way over time. I know everybody's spent. So I just got to spend some time with Sandra, who I know you know very well, and they're in the midst of a ketamine trial, and I think you just went over, so I might have missed a little bit of it. Is it safe to say if CHAMP1, if you knock out CHAMP1, is ADNP expression reduced?
I don't think we know that.
What'd you say?
I don't think we know that.
We don't know that. Okay. I'm not sure if, Dr. Harkin-Ersten, if we did the RNA-seq data, can we confirm that the ADNP was reduced? Okay. So would it be possible in the future or at some point to maybe see if ketamine would also, and I know it was an autism drug, not just something for ADNP. Is that a plausible theory?
Yeah, I think it's more plausible than you think, because I don't think, I'm not convinced that the effect of ketamine in ADMP syndrome is because of the ADMP loss. I think it's this very low dose. The hypothesis of why it was tried was based on some changes that ketamine induced ADMP. But I think the reality is that the impact of ketamine on stress and things like that was such that reducing some of these things that anybody could be suffering from actually improve behavior. So I think it was non-specific, which is not a bad thing. Aspirin is non-specific. We don't take it just because it's this very specific thing we have wrong, right? So nothing wrong with non-specific drugs. But I do think that, I think people were very surprised about how ketamine works so effectively. This was an open label study. In some ways, it means nothing until the complete randomized clinical trial is done, which will happen once there's FDA discussion. But if it works equally well under an RCT, I think there's probably a lot of conditions that may show benefit from it.
Thank you.
Long answer to a short question.
Great talk. Thank you so much. I have a question about the biomarkers, right? So we know in genetics there are potentially genetic correlation between the disease and probably also between the syndromes and so on. Do you expect some biomarkers that are similar, so like a sort of common biomarker correlation and so on that can be used, or?
So obviously when we're knocking down 70 to 80 genes at a time across a very large spectrum of chromatin remodelers that are implicated in NDDs, we're looking for shared pathways, and that's in neurons and culture. It's also possible that some of them would overlap in blood, but I'd much rather see that first in neurons and culture, where the neurons are the primary, cell of interest, and then see if it also something similar happens in blood.
Thank you.
Yeah, thank you, Joe. Really great talk. So in terms of the approach of looking at multiple sort of genes at the same time versus when you have a specific mutation showing a completely different epigenetics profile from even another mutation in the same gene. I mean, what is sort of your gestalt on whether we, are with 250 autism genes thinking about, 250 different precision medicine approaches, or 3-4 final common pathways, even though it's from chromosome 22 or chromosome 13 or chromosome 16 or whatever. What is your gestalt?
So I think both are going to be right. I think that most of the genes we're talking about, it probably doesn't matter where the mutation is. It's a loss of function. I mean, obviously, the reality of CHAMP1, it was first discovered as 13q deletion syndrome. So we know that just, a complete loss of one copy produces much of the phenotype. It doesn't mean there won't be differences in some mutations, but I think first pass, we should treat it as a haploinsufficiency, then we can look for shared pathways. That's a very valuable approach, but it's also absolutely going to be the case that we'll also have to dig deep and confirm that multiple mutations more or less look the same biologically before we, I think you have to do both. But I think, you want to think about gene target therapeutics for each disorder, but you also want to think about pathways that are shared that could be targets. But once you think about a therapeutic, you're going to have to take a moment and say, am I really, really convinced that everybody's more or less can be treated equally, or do we need to do further stratification based on mutation type or place in the genome or in the gene? Nobody said it'll be easy, right?
Thank you so much.
Pleasure.
Thank you. Give another round of applause for all the speakers today.